Hello, good afternoon. Today we'll go through discuss regarding the morphology and physiology of the silk glands of a silkworm, which secretes the raw fiber, which is ultimate resource of the textile and silk industries in our India. Here, this is the subunit that we are going to discuss today. The morphology and the anatomy of silk glands. You know, these are the a diagrammatic representation of your silk gland with beautiful colors I have depicted here. The silk of the silkworms is secreted by means of a pair of labial glands. Labial glands. These labial glands are called as silk glands. Okay. This is a structure of the single silk gland, which is containing two labial glands. The silk glands lie ventral to the alimentary canal. Food pipe ki ventral side lo ivi silk glands అనేవి ఉంటాయి. In full grown larvae of the silkworm, these silk glands will occupy most of the body cavity. Most of the coelomic body cavity is occupied by means of these silk glands. The silk glands are tubular in shape. They are tube-like structures with different diameters in different regions. That is why the diagram itself is depicted by means of different colors. Here they are depicted with red, brick red color, yellow and bluish in color, but all are in different sizes and different diameters, and the shape of the silk glands are also different from anterior to posterior region. Now let me come to the each gland has three distinct regions. You should not forget each silk gland is composed of three distinct regions. One is posterior region or posterior gland, which is in brick red in color, and next one is middle, this yellowish portion. This is called as middle silk gland portion, and the anterior one, which is called as anterior silk gland. Here they are shown with different colors. Anterior silk gland is shown with very thin diameter, and it is in violet in color or bluish in color. And next is middle silk gland, which is showing with yellowish color, and the last posterior silk gland is with brick red in color. Okay. Next is, let me discuss all these three regions in different literature. One is the first one, of course we will come from the posterior silk gland here. The same diagram I am showing you in all these slides. It is blunt. It is blunt, highly folded, and this is also a tubular region, but it is showing many foldings. Of course, it is present in the posterior regions of both glands. So it is remain attached to the tracheal bushes of the silkworm. You know, tracheal bushes are the respiratory organs which are there in most of the insects. So they are remain attached with these bushes in case of silkworm. This part of silkworm will secretes fibroin. Fibroin will be secreted as fibrinogen. This fibrinogen, in future, it will be converted into fibrin upon extrusion. After the formation of cocoon, this fibron will be converted into your fiber. Okay, so here they are showing the fibrin. Okay, so this is what the structure they have depicted here. Next one is the middle region, which is in yellowish color. It is most prominent, and of course it is looking most wider than the anterior and posterior regions of the silkworm, and it is wider part of the silkworm and which occupies most of the region of the silkworm. Okay, so it remains folded. It also having 2 or 3 convolutions or folds which resembles the structure of W, isn't it? So W-shaped structure you can easily identify while seeing this picture, and thus it has 3 limbs. One is posterior limb, middle limb, and anterior limbs. So just like W, you imagine W, these 3 limbs can be observed. The posterior arm secretes sericin 1. Here, these are the 2 layers which are present in the fibrin, and the posterior region which secretes the sericin 1, it gets surrounded by means of sericin 2. Sericin 1 and sericin 2 is 2 layers here. You know, they are differentiated by means of different colors here. And sericin 1 is this one, sericin 2 is this one, and the very pale yellowish is sericin 3. The sericin, this sericin again gets surrounded by means of one more sericin layer that is called as sericin 3. So sericin 1, sericin 2 and sericin 3 are being secreted from the anterior limb of the middle region of the silk gland, isn't it? So middle region of the silk gland will secretes 3 types of sericins. One is sericin 1, 2 and 3 respectively. So this middle region of the silkworm or silk gland also act as the reservoir of fibrin where the later gets mature during the storage period. Okay. So next, the utmost part which is present in the anterior region is called as anterior silk gland, which is having very thin anterior region and has a thin diameter too. So it has no secretory lobes like this posterior and the middle silk gland. No secretory lobes will be present, and only transports the assembled silk to the spinneret. Here, this is the terminal part of the silk gland. Whatever the silk will be produced during this posterior and middle region will be transported by means of this anterior silk gland to the spinneret. In the word itself, you have meaning, my dear friends. Let me discuss what is a spinneret. Spinneret is a part of the silkworm which is present in the anterior region of all the silkworms. It is a projection of the median part of the labium which draws the silk out in the form of fine filament. It will spins the fibrin material which is coming out of this posterior and middle region will be spun, and it will be produced like a fine filament from this spinneret. Okay, spinneret, sorry. So the secreted silk comes out as a thread, daram laga din nunchi vastundi, or like a filament as it passes through the silk press, which resembles a typical salivary pump. Chinna pump laga. What is the main function of the pump? It pumps the liquid from one region to another. Likewise, the filaments or the silk fibers will come out by this spinneret. It acts as a salivary pump, isn't it? The 2 filaments coming out of 2 sides are called brims. Likewise, the 2 spinnerets will secrete 2 filaments which is spun together, and these 2 sides of the fibrin is called as brims, isn't it? Now, The sericin looks like gummy in nature. The sericin is a layer of the two brins then bind together into a single filament. That single thread is called as bale. That single filament is called as bale. Now let me come some histological features of this entire gland. Whatever might be the regions of the silk gland, whether it is anterior or middle or posterior regions, it has, when we take out the transverse section of the entire gland regions, might be anterior, middle or posterior, histologically, cytologically, the entire gland has three layers. If we cut here, it is shown in this picture. It has broadly seen two layers, isn't it? The outer one is called as biologically, the outer layer is called as a tunica propria with uniform thickness. And the next we have middle layer that is not distinct in this picture. The middle glandular layer, which is made up of certain glands with gland cells, which increase in size during later instar stages of the larval development. You know, larval development always includes in the increase of their size. Size nu batti larval stage ni first instar ani, second instar ani, third instar ani, fourth instar ani antaru. Later it will become into a moth. Later it will become into an insect. So what I am trying to tell you is, here the middle glandular part of this entire gland or gland cells, which increase in size during their later instar stages. Larva perugutunna koddi ee middle glandular layer size peruguta untundi. And the last one is of course called as, the ultimate inner layer is called as tunica intima. Don't forget, the upper layer is called as tunica propria and the inner layer is called as tunica intima. It has varying thickness. In the anterior region, the gland, it is present in the anterior region of the gland. This layer is very thick and is shed at each molt. Molting ante ఊడిపోతుంది అన్నమాట. అర్థమైందా? anterior region లో లావుగా ఉంటుంది, అది ఊడిపోతా ఉంటుంది. In the another region, the other regions, the silk gland, it is thin and not shed each molt. Mounting కి ఈ posterior region లో, other region లో ఊడిపోదు, front region లో thick గా ఉంటుంది, ఊడిపోతూ ఉంటుంది అన్నమాట. Molting అంటే shedding of the glandular tissue. Now, and of course, this is very, very important. That is presence of, here you watch this diagram carefully. We have discussed regarding the posterior silk gland, middle silk gland which is in W shape, anterior silk gland which is a bluish in color, which has a thin diameter. We have discussed regarding the spinneret, but the prominent structure that is to be discussed is Philippi glands. These are called as Philippi glands, or otherwise called as Leonate glands. These are otherwise called as Leonate glands అని అంటారు, లేదా Philippi glands అని అంటారు. ఈ వీటి యొక్క ప్రాముఖ్యత ఏంటి? What is the significance of these Philippi glands which are just after the spinnerets? That is very, very important. You may get in the examination the importance, the mechanism, the function of the Philippi glands in the silk glands of the silkworm. In the head region of the larvae, a pair of glands are situated which open into the anterior part of the silk gland near its opening into the spinneret. Spinneret ki chala deggaraga anterior region lo rendu glands open cheyabadi ఉంటాయి. వాటిని మనం ఏమంటాము అంటే they are called as Philippi glands or Leonate glands. It is thought that these glands contribute some way, by producing a waxy material to the silk thread, or to lubricate the passage of this silk while coming out of the spinneret. Spinneret నుంచి ఈజీగా స్పిన్ అవుతూ ఆ సిల్క్ థ్రెడ్ రావడానికి ఒక waxy substance ని ప్రొడ్యూస్ చేస్తుంది అని ఇటువంటి వాదన ఉంది. Hence, the Philippi glands are having a prominent role in the secretion of the fibrin. through the spinneret by producing a waxy material. Friends, this is the structure of the silk gland. Definitely you may get in the form of a small question in your examination. And please don't forget to draw the diagram twice or thrice for your practice.